Hello everyone and welcome to Doses Pathology Review, and in today's video I'm going to talk about the mechanisms of cell injury. So let's get started. Cell injury can result from abnormalities in one or more of several essential cellular components, and the mechanisms of cell injury can target the mitochondria and their ability to generate ATP. They can cause disturbance in calcium homeostasis. They can cause damage to cellular membranes, including both plasma and lysosomal membranes, and they can cause damage to DNA and proteins. And all these alterations can be caused by a single injurious agent. Now let's start talking about these mechanisms, and the first one is ATP depletion. ATP is generated mainly by the electron transport chain and oxidative phosphorylation in the inner mitochondrial membrane. ATP can also be generated by glycolysis, and glycolysis is the metabolic pathway by which cells metabolize glucose to generate ATP. There are many causes of ATP depletion. These causes include hypoxia, and hypoxia is the most common cause of cell injury. And we said that oxygen is the final electron acceptor in the electron transport chain, and if there is lack of oxygen, oxidative phosphorylation will not occur and ATP will not be generated. One of the ways by which cells generate ATP in the absence of oxygen is through anaerobic glycolysis. However, the most common cause of hypoxia is ischemia, and in ischemia there is both lack of oxygen and nutrients. So there will be no glucose reaching the tissue to be metabolized anaerobically, and therefore the outcome of hypoxia caused by ischemia is worse than the outcome of hypoxia caused by any other cause. Another point worth mentioning is that tissues with high glycolytic capacity, like the liver, are able to survive loss of oxygen and decreased oxidative phosphorylation better than tissues with limited capacity for glycolysis, like the brain. Okay, what are the consequences of ATP depletion? ATP depletion can result in a reduction in the activity of the ATP-dependent sodium-potassium pump, and the main function of this pump is to pump 3 molecules of sodium out of the cell and pump 2 molecules of potassium inside the cell to create a negative charge inside the cell. And the mnemonic for this is pumpkin. Pumpkin is a fruit, and the spelling is P-U-M-P-K-I-N. So pumpkin, pump potassium in. If there is no ATP, this pump is not going to function. Sodium will accumulate inside the cell, and where sodium goes, water follows, so water will accumulate inside the cell as well. This is going to result in cellular swelling and swelling of the endoplasmic reticulum. Proteins are synthesized by the ribosomes that are attached to the endoplasmic reticulum. So swelling of the endoplasmic reticulum is going to result in detachment of these ribosomes with a consequent reduction in protein synthesis. In addition, if there is ATP depletion, there will be a compensatory increase in anaerobic glycolysis in an attempt to maintain the cell's energy sources. And as a consequence, lactic acid is going to accumulate, leading to decreased intracellular pH and decreased activity of many essential cellular enzymes. All these changes can ultimately result in irreversible damage to mitochondrial and lysosomal membranes, and the cell will die by necrosis. The second mechanism of cell injury is mitochondrial damage and dysfunction, and mitochondria are sensitive to many types of injurious stimuli, including hypoxia, chemical toxins, and radiation. Mitochondrial damage can lead to failure of oxidative phosphorylation, leading to progressive depletion of ATP and cell death by necrosis. It can also result in abnormal oxidative phosphorylation, leading to excessive formation of reactive oxygen species, which can damage the cell. And we're going to talk about reactive oxygen species in a separate video. Mitochondrial damage is often associated with formation of channel in the mitochondrial membrane called the mitochondrial permeability transition pore. The opening of this channel leads to the loss of mitochondrial membrane potential and pH changes, further compromising oxidative phosphorylation. The mitochondria also contain several proteins that, when released into the cytoplasm, they tell the cell that there is internal injury, and they activate one of the pathways of apoptosis. The third mechanism of cell injury is influx of calcium. Calcium inside the cell is normally maintained by ATP-dependent calcium channels at concentrations as much as 10,000 times lower than the concentration of extracellular calcium or of sequestered intracellular mitochondrial and endoplasmic reticulum calcium. So the concentration of free calcium in the cytoplasm is very low. ATP depletion can affect the function of ATP-dependent calcium channel, and because the concentration of extracellular calcium is 10,000 times greater than the concentration of free cytoplasmic calcium, calcium will move down its concentration gradient. This will increase the concentration of free calcium inside the cytoplasm. Ischemia and certain toxins can cause an increase in the cytoplasmic calcium concentration as well. So, what are the consequences of this? Increased cytosolic calcium activates a number of enzymes. These enzymes include phospholipases, proteases, endonucleases, and ATPases. Phospholipases cause degradation of phospholipids of the cell membrane, resulting in membrane damage. Proteases break down both membrane proteins and cytoskeletal proteins. Endonucleases cause DNA fragmentation, and ATPases cause ATP depletion. In addition, increased intracellular calcium can increase mitochondrial permeability. This can cause leakage of mitochondrial proteins, activating one of the pathways of apoptosis. Increased cytosolic calcium can also cause direct activation of caspases, which are proteases that trigger apoptosis. So, in this video, we talked about ATP depletion, mitochondrial damage and dysfunction, and influx of calcium. And in the next video, we're going to discuss the remaining mechanisms of cell injury.